Good morning, everybody. I'm Michał Grabowski, and I have a pleasure to present on the biodiversity of Poland and DNA data together with Julia Pawłowska from University of Warsaw. Julia will speak for herself, and I'm the representative of the national node of IBO in Poland, and also something we try to build as Polish Barcode of Life, and representative of the Horizon Europe Biodiversity Genomics Europe, which I will also talk about shortly. So basically, I see that the audience is heterogeneous, so our presentation will also be quite heterogeneous, because I'm sure that's, some of you have experience with molecular data, some of you have not. So there will be a bit of introductions and a bit of actually a bit of explanation about what was just told about, meaning the Barcode of Life data systems and so on. Okay, so basically the problem we have is biodiversity, because we still don't know how much of biodiversity we have all over the world. And there are estimations from 10 millions, even up to trillions, if we account for microorganisms. But of course, nobody knows what are species in microorganisms. So basically, so far we have around 2 million of species described. And even if with this most conservative estimation, we still have 8 million of species that need to be discovered and described in scientific names, with scientific names, and it will take some half of a thousand of years to do it. So we need some solution. So basically, Paul Hebert, who is an entomologist from Canada, and I have an impression that entomologists are moving spirit behind the biodiversity research, came with this wonderful idea that species can be somehow tagged with barcodes, like the products in a shop, but it can be done with DNA. So basically, he proposed a standard section of a mitochondrial gene, cytochrome oxidase subunit 1, which is a bit more than 600 base pairs long, that can tag every single species, or at least of metazoans. So basically, that's how it looks. We have 4 letters of the genetic code, and if we translate that to 4 different colors, we have a barcode. So indeed, extracting DNA from an individual, we can sequence this piece of DNA, and we can see, what is the arrangement of these letters or of the colors. So that was the original idea. Of course, then it developed, because it appeared that for different taxa, for different branches of the tree of life, there are different markers that are better than CO1. So we have a whole arrangement for plants, for fungi, and so on. Sometimes it has to be more than one sequence, because one sequence is not enough. And that's a general pipeline of how it's all organized. So we need to collect a specimen, we need to sequence the DNA, and then there is a whole thing with managing the data. And we have these DNA repositories, reference libraries, and Barcode of Life data system is, I think the first one and the biggest one for metazoa. And I will talk about that a bit later on. What is important at this stage is that at the very beginning, in 70s, when the sequencing of DNA started, it was very costly and very slow method. And then with the technology development, I cannot manage with this pointer. Okay, I will do without it. So with the technology of development, we went to high throughput sequencing that allowed to produce millions of sequences at a very short time. So per sequence it was cheap, but all over it was actually expensive. But recently, I would say like in 2014, we entered the new era. It's Oxford Nanopore technology. So we have a sequencer which is of a size of a pen drive, and actually it allowed us to downscale this high throughput sequencing in such a way that we can sequence we don't have to sequence the whole millions of individuals at once to get it cheaper. We can sequence thousands, which is a reasonable solution. So, and of course, the drop down of costs. I mean, look how dramatic this drop down is. So now everybody, every institution, and not even in the rich country, can afford to sequence, to produce these genetic tags for species. And now it's time for Julia.
Thanks. We decided, like, for the purpose of this meeting, to show you what GBIF knows about DNA-related species occurrences from Poland. So we know that right now there is more than 70,000 occurrence records from Poland having either DNA-derived data extension or associated sequences. So it would be good to know how it happened and what are the from where the sequences are coming from, because I guess that the majority of collections are not sharing these sequences with GBIF, so you may be kind of surprised how it happened. For taxonomy, what the sequences are representing, they're representing pretty good variability of organisms we have in Poland. I'd say it's way more representative than for occurrences. So, like, you cannot see domination of birds in here, for example. And we have bacteria that are, and we don't have, like we have in sequence-derived informations, we have bacteria that are not present in our occurrence data sets that we are sharing. So if we try to see the origin of this DNA-derived biodiversity information, the highest number of sequences is coming from this INCDCs, so International Nucleotide Sequence Database Collaborations. These are gene banks. This is European Nucleotide Archive and gene banks. It means that if in your institutions you have researchers who are publishing their sequences in gene banks, and majority of them are doing this because it's mandatory. If they want to publish something, they publish their sequences in there. And if they put geographic information as metadata in the sequence, then it will be shown in GBIF. And so majority of those data are already in there, but they're very, very gappy. And this is something that you should potentially discuss inside of your institution, how we can improve about it. So this is how this data from gene banks looked from Poland in near 2000. Here you have 2014 and 2023. So you can see that, like, there is increase of this data, but it's not kind of really huge amount of data sets. So we can also potentially improve in here. This data sets, yeah, I told you that like in GBIF, you can see those records from gene banks where you have information on locality. But having your GBIF background, I would say, or collection background, you would expect coordinates or something. This is not the case for majority of those data. So we don't have coordinates. The only thing we have is country. And it is already treated as geographic information, so you can see them in GBIF, but the country is really, really high level of geographic accuracy. So if you are in touch with, like, researchers at your institutions who are publishing their sequences to gene banks, please draw them their attention to metadata. They probably have coordinates for these occurrences, especially that a lot of these occurrences are coming from herbarium specimens, actually. So they are publishing sequences from herbarium specimens, but they are not giving coordinates that quite often you have on the specimens. So this is something where we can super easily improve. And like, by curiosity, I was checking what are the specimens which have really, like, good quality metadata. And this is like, majority of these occurrences are just simple, like, barcoding sequences. So someone is taking specimens, barcoding, and here we have the record. But actually the one that has the most complex metadata was specimen coming from, whole genome sequencing. Because if someone is sequencing whole genome, then the metadata are super carefully prepared and published. So also this data from whole genome sequencing are right now visible in GBIF, and they are treated as occurrence data. So we should also have it in mind if we're thinking about biodiversity. This will be of particular importance, for example, for prokaryotes or bacteria. And finally, only recently, we have also eDNA datasets. This is the first eDNA data sets from Poland we published through MDT, so metadata toolkit Joe presented at the beginning. And what you can see here is the number of occurrences which is coming from such one data set. It's like 11,000 occurrence records coming in one bench from one data set. And the second one, which is about to be connected today or tomorrow, is 27,000 species occurrences. So this is something that will change the landscape of occurrence records we have from the country. And we should be somehow prepared to include this data in our biodiversity analysis and working with them. So this is the meta-barcoding data toolkit. We'll tell you a few more words about it tomorrow, but today we'll rather focus on, I will not go in details, we'll go back to BOLT and IBOL, which is, which is like also on the list of this top molecular data publishers from Poland.
Okay, thank you. So eDNA is a fantastic tool, but to use it, it's best to have a reliable reference library, meaning that we have a library of sequences that are identified taxonomically in a reliable way, preferably by specialists. So for that, this part is basically crucial. And here it comes, the amount of data we have at this moment from different parts of the world, different parts of the world. So, to use this information in a proper way, we have to need a complete record for a reference library. And that's a case of a red admiral butterfly, common butterfly in Europe. And that's how it looks, the typical page of, well, the optimal page for a barcoded specimen in IBOL. So basically, that we have an image, we have all the identifiers, including the QR code that we can scan and that we can get all the information about this specimen. We have all the data sets that this specimen appears in the whole database. Then also we have the taxonomy, okay, and we have something, oh, Jesus, here's, yes, we have something which is called BIN, barcode index number. So, meaning that all these sequences that are in BOLT, they are automatically bioinformatically clustered based on the genetic distance and also based on the discontinuities in the genetic distance between these clusters. So, meaning that even if we don't have a name associated with a specimen, it's not a waste of time to put it there, because it will be clustered anyway. It will have this barcode index number, which also may have its digital object identifier, and it can be treated as a rough representative of species, even if we don't know yet what species it is. Then also we have all the information about the sequence, also with a QR code and all the metadata that we need, including, of course, the occurrence record, which is very important because then these records from BOLT are fetched by GBIF. So, and a part of the name, we also have this barcode index number. So, and also in GBIF, we can just see where this barcode index number is distributed all over the world. And here we have also a scientific name associated with it. But if we don't have, and then at some point we have a specialist that identifies an individual from this barcode index number and gives it or associates it with some already existing taxonomic name, then out of a sudden we have the distribution of a truly non-species, which is a very beneficial thing. And of course, there are many applications of DNA barcoding, not only for science, but also in all kind of human business and activities. We'll not talk about that. But one thing that I would like to draw your attention to is ecotoxicology. So quite recently, with colleagues from different countries, we did this analysis about presence of cryptic species, so cryptic species that cannot be identified based on morphology in different groups of, let's say, invertebrates, terrestrial and aquatic, and vertebrates, also aquatic and terrestrial. And it appears that more than half of invertebrate species that are used as model species in ecotoxicology are in fact groups of cryptic species. And if we don't know exactly with which cryptic species we are working, then the whole results of the ecotoxicological tests can be messed up. And one such example is, for example, the amphipod, Gammarus fossarum, that we were studying. It's distributed all over Europe. We found out not only with DNA barcodes, but with different evidence, including hybridization experiments and so on, that it is composed of many different lineages that are representing different species and also having different ecological niches. But to identify them, it's possible only with DNA barcodes at this moment. Of course, we have all these other technologies which are developed based on DNA barcodes, like metabarcoding, eDNA, that is very suitable in studying the ecological interactions, also in ancient ecosystems, which is very important. But okay, so we mentioned the International Barcode of Life. That's a global enterprise that was founded by Paul Hebert, and it's still working, and it has let's say the vision at least until 2045. Now we are in this scale, which is, let's say, eyeball bioscan phase with more than 30 countries from all over the world. And Paul Hebert is a very enthusiastic person and very optimistic. So during this organizing meeting of the eyeball bioscan phase, he told, we are going to barcode the planet. So, well, knowing Paul, I believe it's possible. So basically, the ambition of eyeball until this 2045 is to establish like a global monitoring system of biodiversity based on DNA barcodes, where every species is documented with the barcodes, that also we can study the interactions between species, and we can also monitor the state of biodiversity in different parts of the world. In Europe, we have now the eyeball Europe, that together with the European Reference Genome Atlas are working now within the big project. It's 23 million euro project, Biodiversity Genomics Europe, which operates from 2022 until 2026, and there are 34 partner institutions across 20 countries, and our university is one of them. And what we do, so basically we try to enhance the biodiversity research with molecular methods, so to build the capacity to upscale the production and also to enhance the application of genomic tools to biodiversity. The project is composed of different work packages that are covering all these, let's say, workflow that I presented to you before, from field through sequencing, applications, and so on. And within this project, we are trying to build something which we call the Polish barcode of life. There is Austrian barcode of life, Norwegian barcode of life, German barcode of life, and so on. So we already started to think about it in 2019, but then there was a pandemic and we couldn't do much. So since the project was launched, we are working with this, and we associated this idea with the Polish GBIF node and with this Polish Biodiversity Information Network, actually thanks to Piotr and Julia. So basically, now we have this financial support, which is fantastic, because that means that we can really organize something, let's say, more meaningful. But anyway, we started to work extensively with different institutions, but also with citizens, scientists, and stakeholders in terms of barcoding activities. I will also tell a bit more in a moment. And within this project, we generated quite a lot of DNA barcode data, and we can basically divide it into these 4 different, let's say, groups. So meaning the data for building the reference library of Polish invertebrates, mostly. Then also a project of the pollinators. These are the red dots, which are covering like the entire territory of Poland, and it's actually this sampling stations with Malez traps are distributed all over Europe. So in agro-saisons and also in ornamental gardens to see, you know, what are the pollinator communities. Also the high mountain sampling, which I will just say a few more words about it. This sampling is located in the Tatra Mountains National Park, and also the dark taxa. So this is exactly the case. These are all the gray dots where we don't have the taxonomic identification first. We just have the barcodes, and then we are trying to find out what portion of these barcodes can be identified via reference libraries. And then also with this portion that cannot, we are trying to gather taxonomies to work with it and identify it. So here is the high mountain sampling. So there are 8 mountain ranges all around Europe, from Greece and Portugal to Norway. So the idea is to have 5 different altitudinal zones with paired Malez traps and sample through the whole season since early spring to late September. And we did it here, like in the valley from Zakopane to Morskie Oko and then a bit higher. So that was done last year with an immense help from the Tatra Mountains National Park. So we organized also several workshops with them, not only for their scientific staff and rangers, but also for the other national parks, just to illustrate and to teach them how these genomic tools can be used in the routine work of the national parks, which we got a very huge interest from all those institutions. So this collection is already done and processed right now. This year we did the pollinators and so on. So basically, when we started in Bold, there was not even 5,000 sequences of DNA barcodes from Poland, and representing around 1,000 named species and these 800 beans. So right now we are here. Well, generally it's mostly for metazoans. So majority. So altogether we have like nearly 20,000 sequences, and around 18,000 of them they are metazoans. So you see, it's like 4 times more at this moment. By the end of BG project, I think we will have around 30,000. So right now we have 2,500 beans and 1,600 species, and that's not the end. Of course, if you see at the composition of all this data set for metazoans, of course you have insects, mostly dipterans, and some lepidopterans, coleopterans, and malacostracans. That's because we are working with malacostracans. So we pay attention to that. But basically, much of this is without taxonomic names. I mean, there is identification to genus, to family, but especially with flies, it's difficult to identify that to species. So we are also trying to overcome this problem. So for some of the taxa, like orthopterans, this is actually quite coherent group. There is a citizen science group which is having this online atlas of distribution of Orthoptera in Poland. We are closely working on them. So thanks to them, we have now all the species of Orthopterans from Poland reliably identified, barcoded, and so on. But for dipterans, we are organizing taxonomiton this spring. So we are gathering all the dipteran specialists at our university to work with this collection, where we have this dark taxa, species without names but barcoded, and also to teach the young generation. Because, as you know, there is the taxonomic crisis, so crisis also in number of taxonomists. So we try to overcome that. And last but not least, this BG project is ending next February, but already there is a outlook for the new one. There is a new call for strengthening taxonomic approaches for biodiversity. So both communities, the IBOL Europe and ERGA, they have a call for expression of interest, and institutions can be associated or part of this consortium on very different ways. So also in production of data and also in managing the community. So depending on the needs and so on. But anyway, you are welcome to look at it and see if you can find a place for you there. So if anyone is interested in this part of the activity, the BOLT and barcoding initiative in Poland, so please either contact me or visit our website. Thank you.